All right, ladies and gentlemen, this is going to be our next lecture of the chapter, and we're going to look at some different types of transport. And what that is just some different ways that things can either move into the cell or the way that things can move out of the cell. And there's some different methods that we're going to look at in this lecture. So first of all, we're going to talk about what the cell membrane is sometimes referred to as, and that is what's called the fluid mosaic model. So we're going to go ahead and break that down. When we talk about the cell membrane being fluid, okay, fluid just means that the cell membrane is very flexible, and the phospholipids that make up the cell membrane kind of move around and float around and switch places regularly. So that is going to go ahead and explain the fluid portion. Now, when we look at the mosaic portion, okay, mosaic is actually can be a description of a piece of art. And if you've ever looked at a Spanish mosaic, what a Spanish mosaic is it's a piece of art that is comprised of a bunch of different little itty bitty tiles that then make a picture. So the reason we can refer to the cell membrane as being a mosaic is because we have so many different components that make up the cell membrane. Think about what we talked about in the last lecture. We have all of the phospholipids, we have all of the different proteins that are present, and we also have those carbohydrate chains. So there's a lot of different components, a lot of different pieces that make up the cell membrane. So that's where we get the idea of it being called a mosaic. Now, the fluid mosaic model, or the cell membrane, helps explain how things can leave the cell or how they can come into the cell. And that's how we're going to go ahead and spend the rest of our time looking at this lecture, just kind of looking at how things come in or out, either moving through those proteins or moving through those phospholipids. Now, some important vocabulary that we need to go ahead and look at for this chapter. We're going to talk a lot about concentration. When we talk about the concentration of something, we're talking about how much substance is in a certain volume of a liquid, okay? And you can think of like hot chocolate or Kool-Aid. So you can have hot chocolate that's super, super concentrated, meaning that there's a whole lot of like the hot chocolate packet mix mixed into your water or your milk, okay? And it's super chocolatey. You can do the same thing with Kool-Aid. You can add a whole lot of the Kool-Aid powder into your water and make it very, very concentrated, okay? Or you can have the opposite situation where it's kind of more diluted and there's really not that much of the powder mixture in the liquid. So you kind of get like that faint taste of Kool-Aid or like watered down taste. And that just means that there's really not a high concentration of the Kool-Aid in the water. Now, when we look at a solution, there's going to be two substances. There's the solute, and that's what's going to be dissolved into the solution. So that would be like the Kool-Aid powder or the hot chocolate powder. And then you have the solvent, and that's what's going to be doing the dissolving. So that's typically a liquid. Usually we think of water doing the dissolving in most cases. But what we're going to look at is how the concentration of the environment outside the cell might be different than the concentration inside the cell, and how that might cause some things either to move into or to move out of the cell due to those differences in concentration level. Now, what we're going to first look at is what's called passive transport. Okay, so there's two types of transport, passive and active, and we're going to look at passive first. Passive transport is when we have transportation of substances either moving into the cell or out of the cell without using any energy at all. So this is super, super important. Okay, it is called passive because it's not needing any energy. It just kind of passes on through the cell membrane. And again, that could either be going into the cell or out of the cell. But either way, it's going to transport a substance, okay, without needing any energy at all. Now, the way that this moves is it's going to move from an area of a higher concentration to an area of lower concentration. So if we look at our model here, okay, and we go ahead and we imagine that this little divider is like our cell membrane, okay, on the outside of the cell here, okay, you can probably clearly see that there's a lot more of these purple molecules, okay, than if we were to look at what's on the inside of the cell. So when we talk about the concentration, okay, this area has a much higher concentration of these purple molecules compared to inside the cell. This has a much lower concentration. Now what's going to happen in passive transport is the substance is going to move from an area of high to an area of low. So many of these purple particles are going to start moving across the cell membrane, and this just naturally occurs because they're kind of bouncing around all over the place and deflecting off of each other. And they're going to start moving over to the side that's a lower concentration. Now, at some point, they're going to reach a state of what's called equilibrium. Okay, equilibrium, just like it sounds, okay, means equal. So once there's an equal number of particles on both sides, okay, then that cell is at equilibrium. Now, that does not mean that particles aren't going to continue to move across the cell membrane. It just means that they're going to keep moving at an equal rate. So if we see a couple go inside the cell, we're going to also see a couple more move outside the cell. Okay, so once a cell is at equilibrium, it does not mean that everything stops moving and nothing passes across the cell membrane. It just means that it's moving now at an equal rate. Okay, however much goes into the cell is also going to be leaving the cell. So we stay at that equilibrium until something in the environment may cause it to go out of equilibrium. Okay, but either way, passive transport does not use energy, and it's going to move things from an area of high over to an area of low until they reach that state of equilibrium, and then everything's going to just keep moving at equal rates in and out. Now, diffusion is a perfect example of passive transport. That's one of our main types of passive transport. Diffusion is where we see substances move from an area of high to an area of low concentration. Okay, so if we look here and imagine that this is our cell membrane, okay, we obviously have a very high concentration of our green particles here, and they're going to go ahead and be, again, bouncing around and deflecting off of each other, and because there's so many of them, they're going to be bouncing and hitting each other much more frequently, and they're going to cause them to start to deflect over to the cell membrane and over onto the low concentration side. Okay, eventually, so much is going to be moved over to the low concentration side that it's going to get to that state of equilibrium, where you have even amounts on each side of the cell. Okay, and again, just because it's at equilibrium doesn't mean that things aren't moving anymore. You'll still see some of those green particles actually move back over to that side, and you'll see some move back over to this side. It's just going to be at an equal rate. Okay, things still move when we're at equilibrium. It's just going to be at an equal amount. Okay, so high concentration to low concentration. So if you look at that diagram, what you see happening, you're going from an area of high concentration and you're going to see it diffuse out, okay, until it's covered the entire area. This is the same idea as when if somebody were to spray perfume down the hallway and then you start to smell it all the way across the other side of the hall, okay? Those particles from that perfume actually start diffusing throughout the air and you start smelling it in different areas before where it originated. Okay, diffusion is just the movement of things from a higher concentrated area to a lower concentrated area. Now, things that can affect diffusion, how quickly diffusion is going to occur and how quickly we get to that state of equilibrium. So temperature is a huge factor. The hotter the substance is or the hotter the environment is, molecules are going to move faster and diffusion is going to happen a lot faster. The colder it is, molecules are going to move a lot slower, so it's going to take longer for diffusion to occur. Okay, if we had a beaker that had some warm water in it and we had a beaker that had ice cold water in it and we dropped some food coloring in there, the food coloring would diffuse much quicker in the warmer water to color the water that color versus in the ice cold water. It wouldn't diffuse as fast. Also, something that can affect how quickly diffusion occurs is agitation or stirring. Okay, when you go ahead and dump a packet of Kool-Aid in your water or Gatorade powder or hot chocolate, you don't just stare at it. You typically go ahead and grab a spoon and stir it so that it gets evenly distributed. Okay, so when we stir something, it causes that diffusion. It causes things to occur quicker and get everything kind of spread out much faster. The last thing that can affect diffusion is the concentration. So how much of the molecules are needing to diffuse. The more molecules there are, the more bumping around and deflection will occur, and the faster you're going to see those molecules diffuse. Okay? The less molecules there are, they have more space to kind of roam around, and they're not going to bump into each other as much, so it might take a little bit longer for diffusion to occur. All right, now the next type of transport is called facilitated diffusion. So if we facilitate, okay, the word facilitate means to help. Okay, so facilitated diffusion is still diffusion. We're moving things from a high to a low concentration. It doesn't require energy, but we're getting a little help from something. Okay, and that's something that's going to be helping us are proteins. We talked about in the cell membrane how there are a lot of different proteins in the cell membrane, and those are what are going to help move those substances. So not everything can go ahead and cross through the phospholipids. Okay, so not everything can get through the phospholipid layer, and instead we might need to go ahead and use some protein channels or protein carriers to move those things across the cell membrane. Now, these are two examples of proteins that you would see in your cell membrane. Both are going to help with facilitated diffusion. Again, no energy is needed. This is passive transport, and it's going to move things from an area of higher concentration down to an area of lower concentration. But again, it's just with a little bit of help from a protein. Okay, so we've got that high concentrated area to the low concentrated area. But it's just instead of them moving through the phospholipid bilayer here, they're going to use that protein to help them. Now, the last type of passive transport is osmosis. Osmosis is simply the movement of water. When we talk about osmosis, we are only talking about water moving across the cell membrane. Okay, it is water moving still from high to low. Okay, so we aren't changing the concentration. It's going to go from high to low, but we're no longer talking about any substance. We're talking about just water movement. So if we look at the diagram here, okay, these guys are water molecules, and obviously this side, this left side, we have a much higher concentration of water molecules. So we're going to see those bouncing around and bumping into each other more, and they're going to start deflecting and crossing over the cell membrane to go to the area of lower concentration. So sometimes water we're going to see move, especially if certain particles or molecules are too large and they can't move themselves, and we want to get the two concentrations to equilibrium. We can go ahead and move some water around to get those concentrations in those environments to equilibrium. Okay, think about if you added too much Kool-Aid powder and it's way too concentrated. You can't take out any of the Kool-Aid powder because it's dissolved in the water already. But the one thing you can do is you can add some more water, okay? And that could go ahead and help fix that concentration of that Kool-Aid. So it's the same thing. Sometimes the outside cell environment and the inside cell environment, they're not at equilibrium. And sometimes our particles are too big to move. So instead we start moving the water around to try to even out the concentrations. And that's what osmosis is. All right, now, sometimes when we see the movement of water, it's due to different environments, and that can cause our cells to either lose water and shrink. It can cause them to gain water and expand, similar to what we're going to be seeing in the egg lab that we've been doing in class. Okay, and there's different environments. So a hypertonic solution, if a cell is in a hypertonic solution, that means that water is going to move out of the cell and it's going to cause the cell to shrink. Okay, and what that means is that there's actually more water, there's a higher concentration of water on the inside of the cell, and because in osmosis water moves from high to low, we're going to see that water leave the cell and it's going to cause the cell to shrink or dehydrate. Now, the perfect scenario is if it's in an isotonic solution. Isotonic actually means we're already at equilibrium. That's how you can kind of think of it. There's no net movement of water, so there isn't water just leaving the cell or just going into the cell. Instead, it's going to be at equal amounts. So the amount of water that's leaving is also going to be the amount of water that's coming in. So there's no change in the cell size. Everything's kind of already at equilibrium. The last environment is what's called a hypotonic solution. So if a cell is in an environment that's said to be hypotonic, what we're going to see happen is water is going to move into the cell, and that's going to cause the cell to expand. So that just means that the higher concentration of water is outside the cell, and because in osmosis water moves from high to low, we're going to see that water move into the cell, causing the cell to gain water and expand in size. So those are kind of three different environments that cells can be in that causes osmosis and can cause either a cell to shrink, cause a cell to stay the same, or cause a cell to expand in size. Now, this can be good and bad. So sometimes, like in our red blood cells, if a cell is in a hypotonic environment too much, it can actually cause the cell to burst because it's going to take on too much water and can actually cause it to burst, and that can cause issues. Also, if a cell is put into a hypertonic situation, it can become too hydrated, shrink and shrivel, and actually die and cause a lot of issues as well. So we want to make sure that our cells are in that more isotonic environment where we are able to go ahead and keep them kind of in that perfect environment where the amount is coming in and going out at that equilibrium rate. Now, the next type of transport is opposite of passive. Active transport, like it sounds, active requires energy. So this is where we're going to transport things across the cell membrane, but we're now actually going to have to use some energy to do it. And the reason that we need to use energy is because we're going to be going from an area of low to an area of high concentration. Now, the biggest reason that this is requiring energy, again, is because we're actually going to be moving the opposite direction than what we have been talking about. In passive transport, we've talked about going from high to low, and that's natural. That's what we should be seeing, and that's why it doesn't require any energy. However, in active transport, we're going to actually be trying to move things from a lower concentration the opposite way to a higher concentration. So we're actually trying to get everything over to the really high concentrated area. And because those particles and things don't want to normally move that way, we're going to end up using a little bit of energy to do that. Okay, so that's what active transport is. We're going to use some energy because we're going from low to high. We're going against kind of the grain as to what we should normally be moving in the direction. Now, there's a few different types of active transport. So we're going to use a transport protein for some of them. And that's going to be, again, like those proteins that we see that are within the cell membrane. And that's going to move some smaller molecules, and we use some ATP, some energy in those proteins to move those things. Okay. Two other types of active transport are exocytosis and endocytosis. These are to go ahead and move larger particles either into or out of the cell, and we're going to look at those a little bit more on the next couple of slides. Now, exocytosis is where we release things out of the cell. So think of EX, that prefix, okay? Think of exit. So things are exiting the cell. So if this is the inside of the cell and this is the outside of the cell, what we're going to do is we're going to see large molecules that get released. They kind of fuse with the cell membrane here and release it out into the outside environment. That is going to require a little bit of energy to do that, and that's why it is a version of active transport. Okay, this is just an actual real-life visual from a microscope showing exocytosis. So we're looking at This part here, the arrow's a little bit off, but this part here is the actual stuff that is going to be fusing with the cell membrane and releasing the contents of it. So we call these vesicles that carry the material, and then the outside part of the vesicle fuses with the cell membrane, opens up, and lets everything out of the cell. Okay, so again, exiting exocytosis. Now, endocytosis, that prefix N, just think entering. Things are going to be moving into the cell. So in endocytosis, we're going to have those particles enter and move into the cell. And again, this is active transport. It's going to take some energy to move those larger particles into the cell. And this is a really nice visual of actually a part of the cell membrane coming out to go ahead and grab this food particle and bring it back into the cell. So whenever we're talking about endo, it's usually going to be larger molecules, which is why we need to use some energy to move those things in. But we can think of it kind of how the cell gets its different nutrients and resources in its food. Now, when we look at this endocytosis, this is dealing with our immune system. So anytime our immune system cells realize that something is foreign in our body, like a yeast cell or a virus or bacteria, what that immune system is going to do is it's going to go ahead and kind of wrap around it and take it in to its cell and destroy it so that we don't get sick. So our immune system cells are really, really, really key with doing endocytosis to help break down and kill those things that aren't supposed to be in our body. So this is a really, really cool visual that, again, was taken from a really fancy microscope, and we can get these really cool pictures. So this blue portion here, this is the immune cell, okay, and then this is the yeast cell that the immune system is going to try to kill. So the yeast is going to get engulfed and picked up by this immune cell that's kind of wrapping itself around it, and it's going to pull it into the immune system cell and then destroy it so that we don't get sick. All right, so that's it for the transport lecture. Make sure you really look over passive transport and the different categories of that, looking at facilitated diffusion, diffusion and osmosis, and also active transport and how that requires energy and the different types of active transport, the transport protein, the endocytosis, and the exocytosis. Make sure you ask any questions if you have them.